Good morning to us students. This is MissCarolSamaha, your biology teacher. Today is Wednesday, March 11. We are going to start with a lesson about the fate of synthesized proteins, activity 5, and specificity and the mechanism of enzyme action, activity 8. Please open your books to page 58 to start with activity 5. The objective that we're going to cover in activity 5 is to distinguish between the different fates that the proteins synthesized have. However, the objectives that we're going to cover in activity 8 are 1. Determine the importance of the active site of the enzyme. 2. Explain the steps of transforming a substrate into a product. As a recap to the protein synthesis that we covered earlier, the genes depicted by our DNA are going to undergo the transcription process that occurs inside the nucleus to form mRNA molecules. Then the mRNA molecules exit the nucleus into the cytoplasm. Specifically, they go to specific structures called ribosomes to undergo translation process to form a protein molecule. The protein then undergoes maturation process to attain the globular active form of a protein, and this occurs in the Golgi apparatus in the cytoplasm. Activity 5. Fate of the synthesized proteins. The synthesized proteins can either be functional proteins or cellular proteins. The functional proteins are packed and released from the cell. They have a specific function in the body on a specific target organ. Some examples of functional proteins are enzymes, antibodies, hormones, etc. However, cellular proteins are proteins that remain inside the cell after their synthesis. They have a specific function in the cell, or they are used in building it. Examples of cellular proteins are cell membrane proteins, nuclear membrane proteins, DNA polymerase enzymes, cytoplasmic proteins, RNA polymerase enzymes, hemoglobin proteins, etc. In order to track a functional protein, radioactivity can be used. A very famous example of a functional protein is the insulin hormone. The insulin hormone is a protein molecule that is produced and released by the beta cells of the islets of Langerhans, which are pancreatic cells. In the procedure, beta cells of the islets of Langerhans are cultured in vitro in a medium containing radioactive amino acids such as leucine. 5 minutes later, the cells are transferred to a non-radioactive medium. Cells are then harvested at different times, and autoradiograms are performed where insulin is identified. The black spots shown are an indication of radioactivity. As shown in document A, the radioactivity shows clearly in the cytoplasm. In document B, after 10 minutes of the harvesting, the radioactivity is shown in the Golgi apparatus, which is the site of the maturation of proteins. After 1 hour, the radioactivity is shown in documents C and D inside the vesicles in the cytoplasm. And finally, document E shows the actual fate of the functional protein as the content of the vesicles is released to the outside. The following schematic drawing shows the release of the functional proteins outside a functioning cell. It shows clearly the passage of the proteins through the Golgi apparatus and then to the outside of the cell by the process called exocytosis. Exocytosis is the process of release of molecules to the outside of the cell by the merging of the membrane of the vesicle containing the synthesized proteins with the membrane of the cell. On the other hand, chemical analysis shows that the cytoplasm and the different organelles are composed essentially of proteins. These synthesized proteins are not submitted to exocytosis. The pathway chosen by these specific cellular proteins is shown in the black box in the neighboring document. A very common example of a cellular protein is the hemoglobin molecule that is synthesized inside red blood cells and stays in the cytoplasm of the cell to bind to oxygen molecules, as shown in the neighboring document. And now, please open your books to page 64 to start with Activity 8, Specificity and Mechanism of Enzyme Action. One of the most commonly produced proteins by the cells are the enzymes. The enzyme is a protein molecule that is highly specific to a certain molecule, which we call substrate, and binds to it to transform it into products. Every enzyme has a different active site that binds to one specific substrate. The active site has two functions: to bind the specific substrate using a binding amino acid, and to catalyze the modification of the bound substrate using the catalytic amino acids. Enzymes have a double specificity. They have specificity for the substrate and specificity for the reaction they catalyze. Specificity for the substrate depends on the complementarity of the spatial configuration of the enzymatic active site and the substrate in a lock and key relationship. As shown in the document, not all substrates are able to bind to the active site. Only one specific substrate is able to bind to it. The other type of specificity is the specificity in the reaction catalyzed. An enzyme acts specifically on the substrate by a specific type of reaction. Examples of these reactions could be hydrolysis, synthesis, carboxylation reactions, etc. The following document summarizes the catalytic cycle of an enzyme. In the first step, the cycle begins when the active site of an enzyme is empty and the substrate is available. In the second step, an enzyme-substrate complex is formed when the substrate enters the active site and is bound by weak bonds. In the third step, the substrate is transformed into products in the active site. And finally, in the last step, the enzyme releases the products and the active site becomes ready to accept another molecule of substrate.